How do you say in Flippin Science? We're looking at biotechnology and restriction enzymes.
I'm a restriction enzyme and I'm here to say that I cut DNA in a specific way. If my cuts are staggered, then I make sticky ends, but if I cut straight across, I make a blunt end, my friends. When I make a cut, it's specific and precise. I find my recognition sequence and I make the slice. I was isolated from bacteria, which is not new news. The new news.
Thanks to Sam Moyle for producing this PowerPoint. So today we're going to look at what biotechnology is, so involving the use of plasmids and viruses as vectors, bacterial enzymes and yeast to transfer genes between species. And we're going to look at how particular genes can be selected using probes and removed using restriction enzymes. So we're going to look at what restriction enzymes do. Genetic engineering. What is genetic engineering? It's basically we can take DNA from different sources and combine them into new organisms. This was always seen as a bit of an evil kind of science fiction-y scientists doing bad stuff thing when I was growing up, at least. So here we have a mouse that has a fluorescent gene. So the gene for fluorescence has been inserted into the mouse and particular parts of the mouse are glowing. What's the advantage of this? So we can manipulate DNA. So combining DNA technologies, we can call it recombinant DNA technology. This is where we take DNA from one species and combine it in another species. That's where the recombinant comes from. We can call it genetic modification, or we can just call all of it genetic engineering. We've already talked about how DNA can be extracted from cells. We can read the code on the DNA and we can pinpoint sections of DNA that we want to work with. So we can identify particular genes or particular areas of the genome that we want to work with. And then we can cut those pieces of DNA out using restriction enzymes. Now I've mentioned restriction enzymes before. Let's have a look at how they work. So they're proteins that come from bacteria and they cut DNA at specific regions. It relates to a specific sequence of DNA. So the restriction enzyme wanders along the DNA, finds the sequence that is the one that it's looking for, and then it cuts the DNA at those points. So the enzymes always cut the DNA at the specific places. So this is EcoR1. This is one of the first restriction enzymes you learn about. It cuts the DNA at GAA-TTC. So here we can see a GAA-TTC. The restriction enzyme comes along and it cuts the DNA at that sequence. It's called EcoR1 because it comes from E. coli, Escherichia coli, the bacteria. And we can use this restriction enzyme, even though it's from a bacteria, it's an enzyme, it's a protein. So it can be used for any DNA and it will cut through the DNA at that section. When it does that, it leaves these sections called sticky ends. So it's sections of exposed DNA. You could insert a gene into this area. As long as it finishes off with these sequences here, or the complementary sequence to these ones, I should say, you can chuck a new gene in, and that's one aspect of genetic engineering. So the sequence of bases is called the restriction site. It's usually 4 to 6, and here's the GAA-TTC from EcoR1. Some cut straight across the double helix, and they leave blunt ends, or they can cut diagonally, and that's where we get those sticky ends. The restriction enzymes evolved in bacteria to protect themselves from viruses and other mobile genes that would come in. So by having the ability to cut the DNA up, that means the DNA wouldn't be inserted into the genome, and that would protect the bacteria. So the restriction enzyme, we can use that to cut out the piece of DNA that we're looking at, and we can then use probes or antibodies to identify the specific gene that we're looking for. So I've mentioned a probe before. It's a short piece of DNA, single-stranded DNA or RNA. To use a probe, we need to know the sequence of nucleotides that we're looking for, so the A, C, Ts, and Gs that we're looking for in our gene. And we can do this by reverse engineering our protein. So we look at the amino acid sequence in a protein. We know that the 3-letter codes that make up the codons and the anticodons that go towards putting that amino acid together in a protein, and from there we can make up a DNA sequence. Probe will be a complementary strand of DNA, nucleotides, and it's also radioactively labeled. Now the reason why we radioactively label it is because then we can identify if it has been attached to the DNA, and we can see where that's happened in cells. So here's our probe. It's radioactive, so we can see it using particular photography techniques. We heat up our DNA, we chuck in our probes that are radioactively labeled, and they'll attach to the complementary strands, and we can see where they are on the genome. There's another way of doing this using antibodies. So we're looking for a protein. We inject that protein into an animal, and when you inject proteins into animals, you make antibodies against that foreign entity, so the protein. Those antibodies can be taken out of the blood. You do that using various techniques, and then you can label those using radioactive or chemical labels, these days using fluorescent markers. You have the antibody that's complementary to the protein, and it's labeled in some manner so that we can identify it. Here's step 1 again. So we inject the protein, the animal makes antibodies. Those antibodies can be removed from the blood, and then we label them using radiation or a fluorescent dye. We then take the DNA from the cell, we cut it up using restriction enzymes into fragments, and we chuck that into a bacterial cell. Some of the bacteria will take up the DNA fragments and express them into the proteins related to the genes that we're looking for. If we chuck in the labeled antibody into the bacteria that have taken up the gene want to identify will start to glow because we labeled the antibody radioactively or fluorescently, so we'll be able to see them glowing. We can then take those bacteria that have incorporated the DNA into their cells, and then we can make many, many copies of those. So we can clone those genes, then again, we could use restriction enzymes to cut them out. So we've got our bacteria that's incorporated the gene into its genome. It started making the protein. The antibody will attach in a complementary fashion to the protein that's being produced, because this is complementary to the protein that we started with. And because it's shiny, we'll be able to see that that's happened. If the bacteria aren't expressing that protein, then the antibody won't attach, and we won't see that fluorescing or the radioactive labeling happening. So today on Flippin Science, we talked about what biotechnology is, and we talked about restriction enzymes. That's the Flippin Science Day. Bye.